Okay, so now what we're going to do is we're going to continue with this discussion of signaling, but we're going to move into a discussion of what happens with Euslog-O-Matus. And as you may recall from last time, Euslog-O-Matus has a mating response pathway. So this has to do with the pheromones and pheromone receptors. And that response pathway is primarily a response to these pheromones, and it leads to mating. If you recall, that particular pathway is a MAP kinase pathway. So you've got this pathway we talked about last time with the MAP kinase, UBC4, MAP kinase, UBC5, and MAP kinase, UBC3. In each case, the subsequent phosphorylation of each of these leads to activation of the next part in the pathway. Now, in the case of the mating pathway that responds to pheromones, UBC3 phosphorylates PRF1, and just to remind you, PRF1 is a transcription factor that's necessary for both completion of the mating program and also, in a sense, it becomes a way to start the pathogenic program. It's not really the overall control of the pathogenic program, and that is done initially by the B locus. So PRF1 is able to serve as a transcription factor that affects several different genes and loci. First of all, it's able to act as a positive regulator of itself. And also, there's an upstream activation sequence that is, again, I believe, responsive to nutrient conditions. Or other environmental cues. So we can activate further expression of PRF1 by PRF1 itself, and when phosphorylated, this transcription factor can do a number of different things. So we'll talk about the fact, so we just mentioned that UBC3 phosphorylates PRF1, but another pathway that's involved in phosphorylation of PRF1 is the cyclic AMP-dependent pKa pathway. In this case, the catalytic subunit of pKa is called ADR1, and so when ADR1 phosphorylates PRF1, it serves to activate expression of genes in the A locus. And what that means is that the mating pathway itself, so the ability of cells to mate, is in part dependent on nutrient availability and environmental conditions. And that's what the pKa pathway responds to, is environmental conditions and cues. So phosphorylation of PRF1 will allow PRF1 to potentially activate the genes in the A locus. And it turns out, as we'll see later, that both MAP kinase and pKa have sites on PRF1 for phosphorylation. And so depending upon which sites are phosphorylated on PRF1, that's going to determine what job PRF1 is able to carry out. All right, now, just to bring this into comparison with the pKa pathway that we talked about for Saccharomyces, for this particular system, what you have is, again, an alpha subunit of the G protein. This one's called GPA3. And GPA3 activates UAC1. Just as a reminder, if there are arrows, that means activation. If there's this crossbar, that means inhibition. Okay? So UAC1 is the adenylate cyclase, which makes cyclic AMP. Cyclic AMP is able to bind with the regulatory subunit of pKa. In this particular system, that's called UBC1. Normally, UBC1 would have been bound to the catalytic subunit called ADR1, but in the presence of cyclic AMP, the regulatory subunit is removed, ADR1 becomes free and can phosphorylate those proteins in the cell that need to be phosphorylated, one of which is PRF1. Okay? All right. Now, as I said, both the MAP kinase and the pKa can phosphorylate PRF1, and the phosphorylation state of PRF1 determines what things PRF1 is able to act on. And so what happens is, if PRF1 is phosphorylated by both of these kinases, then it activates by binding to a pheromone response element. It activates the B locus genes, and if this is occurring during mating, so after cells of opposite mating type based on the A locus have come together and a dikaryon is starting to fuse or starting to be formed, then you're going to have a B east from one of the partners in the mating and a B west from the other partner in the mating, and they will come together to form a transcription factor that does two things. One of the things that transcription factor will do, and again, this only happens when you have mating with opposite mating type cells, when that transcription factor is formed and is active, it binds to sites called BBS sites in the A locus and blocks the A locus. So the idea is that when the B locus is active and a functional transcription factor is formed, that's going to block the expression of the A locus. But the other thing that it's going to do is it's going to activate genes that are involved in the pathogenic program. So it's going to allow for stable filament formation and the rest of the steps that we talked about in terms of infection. And what I mean by that is that if you have an active B east, B west heterodimer, so if that's active, then genes involved with initiating the pathogenic program for the fungus are going to be turned on. Okay, question?
What does blocking the expression of the A locus do again?
So in this case, it'll turn off the A genes because they're no longer needed.
Okay.
But there is a delay because one of the things that is in the A2 locus are genes that control mitochondrial inheritance. So this doesn't happen until the cells have actually fused and the formation of dikaryon is about to be initiated. Okay? So when the initial cell fusion occurs, you've got these proteins in both of the cells that are there. And if you have a B east from one of the partners that's different from the source of B west from the other one, then those two can get together, form this transcription factor that's active, and block further expression from A, but activate genes that are involved in pathogenesis. Okay?
Gotcha.
Glue? Very clear? Okay, good. Okay, now, one thing you may have noticed, or maybe you haven't noticed, but there are a number of gene products with similar sounding names. Maybe you could point those out.
The A locus and the B locus.
Well, locus is in both of those names, but what other participants in what we've talked about have similar sounding names?
UBC.
Yeah, there you go. UBC. Yeah, there's UBC4, UBC5, UBC3.
UAC.
Well, UAC is a little bit different than UBC though. right? So you've got at least 3 components, actually 4 components in the MAP kinase pathway. All of them are UBC. And you've got this component here, UBC, in the cyclic AMP dependent protein kinase A pathway. So what does this stand for? Well, this is once again one of those situations where we isolate mutants and we see how that changes the phenotype of these cells. So UBC stands for Usilago bypass of cyclase. So whenever anybody isolated a mutant in this particular gene, what they found was that that relieved mutations in the adenylate cyclase gene. Okay, so let's think about what happens if you interfere with these different steps in the pathway. So what would happen if there were a mutation that wiped out the function of UAC1?
You
Couldn't enter the pathogenic phase.
Right, but or if we want to do it step by step, what would happen in terms of signaling?
No CAMP.
No cyclic AMP. If there's no cyclic AMP, then
There's no split between ADR1 and UBC1.
that's right. And if that doesn't happen, what is not going to happen as far as ADR1?
It will not activate PRF1.
Correct. Right. So that's going to block the ability of ADR1 to do what it normally does. And so then the question is there a way that, and just parenthetically, if this happens, it leads to constitutive filamentous growth. On rich medium. So when they're not starved at all, they're not supposed to be doing this. But the way these cells normally grow is as budding cells. But if you knock out the adenylate cyclase gene, UAC1, these cells, haploid cells, will grow as filamentous, fuzzy structures on agar plates. Okay, assuming that it's rich media, that all the nutrients are available. Okay, so that's weird, right? And what happens is if you get a second mutation, so now if you get a second mutation here, what do you suppose that'll do? What if you knock out now, in addition to this, you knock out this?
Well, if it bypasses the cyclase, then it, like the PKA, like the catalytic ability of PKA wouldn't be hindered.
That's right.
So it would, like the pathway would be restored.
That's right. So getting this second mutation, this one in the regulatory subunit, so now we don't have a regulatory subunit, so we don't care anymore because now ADR1 is able to do its job. And that's why we call this gene a bypassive cyclase, because if there's a mutation in it, then that will bypass the need for the cyclase. Okay? Now, that doesn't mean that that fix is going to make the cells wild type. As we've talked about before, a lot of times there are unintended consequences of doing this, because normally ADR1 is supposed to be regulated, right, by environmental cues. So if you completely eliminate its regulation, then what's going to happen is you may get some unintended phenotypic consequences. And so what this leads to, so if you have, forgetting about UAC at all, but if you just eliminate this gene, you have a constantly active ADR1, and those cells are, so we'll look at it this way. So UBC1 minus cells have constitutively active ADR1, and they are multiple budding. On rich medium. Okay, so they're not exactly back to normal if you bypass the adenylate cyclase. Okay. Now, if that's well, let's go back to how we figured this all out. So is this part clear up to this point? Okay. All right. So the way that this is done experimentally is you ask, what happens when we make a mutation in a particular pathway? And so not we, but the people that studied this initially, which includes Scott Gold and also Regina Commons' lab, what they found was that if this gene was knocked out, you get constitutive filamentous growth on rich media. Okay? And the way that they experimentally did this is they just made mutants. So they made mutants and they said, okay, you've got this new phenotype. What's responsible for that? And the way that they answered that question is they made what's called a gene library. Okay? You may or may not be used to going to the library to check out books, but that was something we used to be able to do back in the day. Now you probably just do everything online, but yeah, back in the day, you go to the library and check out books, volumes that have particular pieces of information that you were interested in, maybe. So a gene library is similar to that, except what's happened is you've broken up the entire genome into small chunks and organized them, ideally organized them into different ways that each can be accessed. And so when you do an experiment like this where you've made a mutant, what you can do is take the entire library, each of which has a different piece of the genome, and put that into the mutant and ask which of those volumes, which of those genes is able to fix the problem. And so when they did that initial experiment, they found that the volume that fixes this phenotype is the gene that codes for adenylate cyclase, which they now call UAC1. Does that make sense? Okay. For those of you who are interested in this, the kind of library that they used was called a cosmid library. It had to do with the fact that Lambda, our friend Lambda, is able to package large pieces of DNA. So if you take the packaging capability of Lambda and then you chop up your genome into the right sizes, in this case you can clone up to, I want to say, 45,000 base pair pieces. What you can do is then you have a whole library of pieces that are about 45,000 base pairs in length. So it's a manageable number. In the case of Usalago, that turned out to be about, I'm going to say about 5,000 clones. But you take all that, you take all those pieces from the library, put it into your mutant, and you ask which one changes the phenotype back. And so from that, we were able to identify the UAC1 gene. Okay. Now, the next thing, of course, is to figure out, so how does the pathway work? So we've identified one component of the pathway, which is the adenylate cyclase gene. So what you do next is you then take UAC1 and you look for revertins. So from this UAC1 minus mutant, let's see, I guess I can erase this part of the pathway, I think, right? Do that. You take this mutant, and what do? We select for, or we isolate revertins. Okay. Now, some of the revertins are going to be cells that have just recovered their UBC1, right? Excuse me. Some of them. Okay, I am not explaining this well. Step back. what we want is to take the UAC1 mutants and isolate revertants. So revertants are going to be budding again. And then once we have these revertants, what we do is we take our genomic library again and we introduce that into the revertants and we ask which pieces of DNA from the genome can now make the revertants go back to being UAC1 minus. Does that make sense? That's a complicated path of events. Should I say that again? Yeah, question.
Why wouldn't you just take wild type and then introduce it? Why would you have to use revertants?
Well, so if you take wild type and you introduce the library, how's that going to change the phenotype of wild type? So wild type is normally budding on rich media. If you introduce the gene library, you're going to introduce different pieces. But which of those pieces is going to necessarily change this phenotype that we're talking about, which is from budding cells? So in other words, what we're trying to do is we're trying to find out how can we correct this? How can we revert this phenotype? And one way would be just to have a true revertant where UAC1 is corrected. But what we're really looking for are second mutations that essentially suppress or bypass this mutation. Okay, does that make sense?
Yep.
Okay, so we've gotten revertants. Some of them are just returning with UAC1, but others have mutations in other genes. And so the purpose of this particular strategy is we want to find out what those other genes were. What genes got mutated in order to create these mutants, these revertants? And the way that we find out which genes they are is by introducing pieces of the genome in to fix their problem. You have a question?
Or I was just gonna say if I could explain it back to you, you could tell me whether I understood it correctly or not.
Okay, that sounds like a good plan.
But essentially you're creating a group of UAC1 minus mutants, and from there you're isolating wild type expressing,
Well, you're isolating things that look like wild type.
that look like wild type.
Right. So that's what we would
And
call
some would be a true revertant in
Right.
that it's been put back to originally how it was,
Mhm.
because statistically that would sometimes happen.
Right.
And then the other ones would just have another mutation that would mitigate the original or the mutated effects.
Yeah, that's exactly right.
Okay. And then from there you're puzzle piecing in little sections to see if you can fix, or if you can see where the second mutation is.
Exactly.
Okay.
So what's going to happen is you've got the revertants, so they're budding, and then you're going to take them and you're going to introduce your library to see if you then return to the UAC1 minus phenotype. And using that process, what they were able to do is identify, first of all, UBC1. So in other words, we already said at the beginning, if you disrupt UAC1, you don't make adenylate cyclase. And so that's going to send these cells into this filamentous growth, okay? Because you're blocking ADR1 all the time, okay? But if you now have a mutation in UBC1, that's going to bypass that because now the ADR1 is active all the time. So the way they figured out that UBC1 was the participant was if you have this mutant that's UAC1 minus and you get a second mutation in UBC1, they now look because ADR1 is always active, they go back to budding. Even though it's not normal budding, they go back to budding, okay? So they're no longer fuzzy or filamentous. If a gene from the library comes in and it's UBC1, a functional copy of UBC1, What will happen is that'll fix the UBC1 mutation that's in this double mutant. Right? So these revertants are presumably, assuming that I can write this down, these revertants are UAC1 minus UBC1 minus. Okay? And that's why they do that. But basically, UBC is this is the question mark, right? Is it UBC1? We know for a fact that that's one example. If you have UAC1 minus and UBC1 minus, they will be revertant. If you take the library and introduce in pieces, one of the pieces that will make this go back to being UAC1 is the piece that contains a wild type copy of UBC1. And so if you isolate that and you identify the sequence or what the gene is, you find out, yes, this is UBC1, which is the regulatory subunit of PKA. Okay? So far, does that make sense? Okay. So then the next question is, so what does that mean about these other things that are called UBC?
It's like in the one like certain area, like if there's mutation, they're also able to bypass like the cyclic AMP step to return back to the UAC1 minus like filamentous growth.
That's right. And it also means that if there's a mutation in them, so remember I said that the revertants were UAC1 minus, UBC1 minus, but that also means that UAC1 minus, UBC4 minus, or UBC5 minus, or UBC3 minus, or UBC2 minus, if any of those other mutations occur in conjunction with UAC1 minus, they'll return to being budding cells. And what that tells you, which is something we already sort of illustrated all along, is I've drawn these pathways, the mating pathway and the PKA pathway, as if they were linear pathways that did not proceed in a more interactive mechanism, but they do. Clearly there's crosstalk between them. At least one place where that's evident is with regard to PRF1. But that suggests that there may be more crosstalk going on between the MAP kinase pathway and the PKA pathway that explains or helps us to understand what's going on with the phenotypes of Usilago. Okay, any questions?
Like in terms of the ratio to like UBCs to UACs, is that like why there's so much more UBCs versus the UAC?
I think it really just is a matter of that disruption of the MAP kinase pathway is going to have an effect on the role that this regulatory subunit has on ADR1. And we don't fully understand why, but we do know that there must be some way in which that MAP kinase pathway is having an effect on the downstream events that lead to filamentation or budding. We just don't know exactly how. So it may not even be directly by an effect on ADR1. There may be some other parallel pathway that affects filamentation or not on low ammonium, on growth medium, that leads to this change in phenotype, whether it's directly related to ADR1 or not. And in that parallel pathway, if you have any of those UBC mutations in the MAP kinase pathway, that's going to allow you to get budding cells even if there's no adenylate cyclase. Okay? Any questions? Okay, so we'll take a break now, and then we're going to switch to PowerPoint slides in just a bit.